If you're an AP Bio student or just taking regular biology in general, there's a high chance that you'll run into this guy, glycolysis. This video, we're going to go over a simplified version that will really take you through the basic fundamental steps of how glycolysis works so that you can get the A on your next test, exam, or even the AP exam when you're studying for it. So, what is glycolysis in the first place? The term glycolysis literally means to split sugar. It's sugar splitting. So glycolysis is very specific to the sugar that it's splitting, and it's splitting glucose. At the state of glucose, a cell can't just use glucose itself in the larger form that it is currently in for nutrients and supplying for energy and overall cellular functions. So we have to break this down. So in order to break down or break apart, you have to be able to destabilize the sugar. And what this means is, in the state of glucose right now is stable. It has good bonds, the bonds are uniform, they are stable in all forms, and there's nothing that's making them want to break apart. And in order for us to make this glucose simpler, right, break it down into smaller steps, you have to make sure that those bonds break so that new ones can be formed. And that's a process, a part of destabilization. In order to destabilize glucose, you have to use two particular substances. The first is ATP. Now, I'm sure you've heard of ATP before, right? Mitochondria, powerhouse of the cell, it fuels, acts as energy sources, right? Things in that nature. In the context of AP Bio, you're going to want to have a deeper understanding of what really ATP is and how it's utilized in a variety of ways in the organism. ATP stands for adenine triphosphate. And essentially, if you recognize the term adenine, that's referring to a nitrogenous base that you probably have seen in DNA. And that's correct. It is adenine from DNA. And ATP, in this state, it's actually unstable. You would think that ATP, right, just like glucose in its original phase, is stable, but it's not. Because of the 3-phosphate that we have here. Triphosphate tells us that there are 3 phosphates, and each phosphate is negatively charged. And referring back to our chemistry concepts, as we know, like charges repel. So these negative charges are going to repel each other continuously. And when something is repelling, repulsion in general creates instability. And that's why you'll see these phosphates are really going to want to break apart from each other, making ATP in that state with 3 phosphates unstable. We want to use the chemical energy that is stored within ATP to remove one singular phosphate, take that phosphate, and destabilize the glucose with it. These phosphates are going to be what brings the glucose to a point of instability. In glycolysis, you're going to use 2 ATP. Therefore, you want to have 2 phosphates, inorganic phosphates, that will go in and destabilize the glucose. The process to which ATP is broken down so that we can have these 2 phosphate ions is hydrolysis, right? We hydrolyze the ATP from ATP to ADP. And this makes sense, right? Because ATP stands for adenine triphosphate, 3 phosphates, and ADP is adenine diphosphate, 2 phosphates. So we effectively just removed 1 phosphate from the ATP. And in this state, ATP is now stable in ADP form. To backtrack, right, we're taking 2 ATP, hydrolyzing them to ADP. 2 phosphate groups are released from the ATP. We send those to glucose. They begin the destabilization process, making the stable glucose start to get to the point it'll want to break apart its bonds and break down. Our goal of glycolysis is, again, to break down the glucose. But there's another factor that's going to play a part in breaking down glucose on top of using ATP, and that's going to be an enzyme called dehydrogenase. Dehydrogenase is going to have coenzymes. Those coenzymes are called NAD positive. There's 2 coenzymes in dehydrogenase. It's essentially just 2 NAD positives. So what dehydrogenase is, right, it's an enzyme. Coenzymes are essentially a part of that enzyme that allow the enzyme to work, right? It's kind of just as the name implies, it's a coenzyme. It works together. It helps the overall enzyme perform the function that it is meant to perform. NAD positive is referred to as an electron carrier. We know that glucose is made up of atoms, right? And in those atoms, what do we know about atoms? They have protons, neutrons, and electrons. In glycolysis, or in general, there's a transfer of these electrons from the atoms present within the overall molecule. What this means is, during the process of glycolysis, after we have initially invested those 2 ATP molecules to destabilize the glucose, we now want to remove electrons. And the way we do this is through dehydrogenase. So what this enzyme does is it removes hydrogen atoms, and when it does so, it gets what a hydrogen atom is, right? It has one proton and one electron. The hydrogen atoms themselves prove very useful to do this because, again, they have one proton, one electron. They're very simple atoms. So it's very easy to remove them from glucose and bring them to the electron carriers, NAD positive. So here's the second thing that you'll definitely need to know in biology, whether you're getting ready for your test exams, whatever it may be. NAD positive, we can refer to this as like a taxi cab. In this form, NAD positive, this tells us that the positive sign tells us that there is actually less electrons, right? More protons because protons are positive, electrons are negative. So whenever you see a positive, that indicates that there's less electrons present because, again, you have more protons outweighing, so creating an overall charge that is positive. In this case of NAD positive, in this current form, it doesn't have anybody in its cab. That means that it can actually accept more individuals, people into the taxi cab to take them across town. Those people would be electrons. In that form, NADH is empty. When we accept an electron, just one, right? Remember, we're taking a hydrogen atom. The first part that we take from that atom is the electron, and that makes NAD positive into NAD. What changed, essentially, is there's no longer that plus sign, telling us that the electron balanced out the protons, and now we've created an overall neutral force. There's still one more thing that we can add to fully max out the electron carrier because, again, an entire organism's cells are always working to maximize the efficiency. In other words, they want to do the most with the least amounts of resources. NAD can then take that hydrogen proton, right, that we initially took from glucose, and it just adds it on like so. So we went from NAD positive to NAD to NADH. And again, we would do that one more time, taking another hydrogen atom out to fill this second coenzyme, a part of dehydrogenase. NAD positive as a general electron carrier, it will be discussed throughout all of your units in biology. It's very frequent, so it's good to really get a good grasp on it in the beginning. So quick overview of taking 2-ATP, hydrolyzing it to ADP so that we can get our 2-phosphates out, taking those 2-phosphates, giving them to the glucose, destabilizes the glucose, gets it ready for breakdown. Then we're also taking at the same time our NAD positives from our dehydrogenase enzyme, and we're removing, through this enzyme, we're removing hydrogen atoms that we are then adding to the NAD positives to make them NADH. At this point, we've filled both of our electron carriers, or taxi cabs, and now the glucose has finally reached its next stage. Glucose breaks down into something that is called pyruvate. Pyruvate is essentially just 3 carbon sugars, and we form 2 of them from glucose. Glycolysis actually makes energy. Now, if you recall back to the initial investment, right, we added 2-ATP to even start this. So once glucose is broken down into pyruvate, there's this side step, and the side step is called substrate level phosphorylation. Basically means that in order to recoup the energy that we invested into glycolysis, and at the same time actually make more than we used, right, to get some actual energy made out of the process. We're taking those ADPs, if you remember, that we originally hydrolyzed from ATP to make glucose more destabilized. The phosphorylation part of the whole entire term refers to adding a phosphate to ADP, and when we add in that phosphate, again, we get what we started with, ATP, right? And this is through dehydration synthesis. We make ADP into ATP by adding that phosphate. And this is the most important part of it, really, is you actually you use 2 of the ADPs that you had originally that you invested from the ATP. And then there's also, you have to remember, the cell has millions of things going on at once. So there's actually other ADPs available in the cell itself. Here's the key thing that you'll really, really want to know. It makes, in total, 4 ATP. Here's the biggest mistake that a lot of people make when they're talking about substrate level phosphorylation and glucose in general. 4 ATP is called the gross output. Basically, all gross means is it's the total amount of whatever was produced. And it doesn't account, or doesn't actually count in how much you spent, or other different expenses. Kind of think of it as money. So if you get a paycheck, right, that paycheck is $500, let's say, but that's your gross amount because that doesn't include the taxes. So once your taxes are taken out, then you get your net pay. And this is the same exact thing with substrate level phosphorylation. We have a total amount that this process makes 4 ATP. And then what we do is we have to account for the 2 ATPs that we originally invested into starting this process, in general. So we take 2 out, and that leaves us with 2 remaining. So we have 2 ATP molecules that are synthesized or made, right, produced from glycolysis as a whole. Now, we can't forget that we still have 2 pyruvates that we got when we decomposed or broke down the glucose. There's lots of different ways that these pyruvates can be used. There's honestly too many to cover in one single video, but the 2 main ones. This also always depends on the type of organism that you're referring to. So we know that there's eukaryotic organisms and there's prokaryotic organisms. Again, different prokaryotic organisms would be more like your bacteria, and then eukaryotic organisms would be more like humans, right, mammals. At this form, there's still a good amount of energy within this pyruvate, and there's enough energy to the point where we could even break this down further, because again, we want to use as much and be as efficient as we can as a cell. So one thing that you'll look into is something called fermentation. The reason why we would want to kind of break these down, we originally, in order to break down glucose, we made NADH. Glycolysis is a cycle. So the cell has to keep undergoing glycolysis over and over and over again. And to do so, the NADH is super, super important, right? Because this is our electron carriers. Without these electron carriers, we can't break down glucose. NADH, we know that we had 2 of those that we fully filled. We can't just let that be, right? We have to basically empty out the taxicab or empty out the electron carriers so that we can get NAD positive, and then that NAD positive can go back on to the initial part of glycolysis and find a new glucose molecule to take away its electrons. And then one way that we can recycle this NADH is through fermentation. Fermentation is basically just referring to the process that a cell can take when it doesn't have oxygen available for it. There's 2 types: alcohol fermentation and lactic acid fermentation. So with alcohol fermentation, what happens is we take our 2 pyruvates that we have, right? And also, super important to note that both the processes within fermentation, they don't make any energy. They are just simply replenishing the NAD positive supply, right? We know that our NADH, it has electrons in it. So in order to recycle it, we have to remove those electrons so that it can become NAD positive again. We take our NADH and we basically give its electrons to the pyruvate to basically reduce it. That's what's in chemistry, it's known as reduction, when you're taking more electrons and you're giving it to a molecule or substance to basically make it more negative. That pyruvate gets broken down into acetaldehyde. In that process of breaking down pyruvate, which is unique to alcohol fermentation, we also make a waste product, CO2. So CO2 basically leaves while we're making our acetaldehyde from breaking down our pyruvate by giving it more of those electrons. More electrons are added to the acetaldehyde and we get ethanol. And remember, we do this twice because we have 2 pyruvates from glucose that we initially broke down in glycolysis. So that's the process for alcohol fermentation, but let's not forget there is one more part of fermentation as a whole, which is known as lactic acid fermentation. So lactic acid is the other option that the cell can undertake instead of doing alcohol fermentation, depending on what it needs, right? It doesn't mean that the cell is going to do both lactic acid and alcohol fermentation, because again, we only have 2 pyruvates. What we're doing the same thing. We're taking the NADH, we're adding it to our pyruvate, and as a result, once we reduce that pyruvate and break it down, so we get 2 lactates from 2 pyruvates by taking our NADH, giving it the electrons, right, and destabilizing the pyruvate, reducing it down to lactate. And then as a result, most important part is we take our NADH and we make it back into NAD positive. If you like this video and you thought that it was pretty informative, make sure you guys drop a like down below and consider subscribing if you want to see more videos about biology, science in general that can really help you in class.